Доктор Алтангэл Нарангэрэл одоо Texas A&M University Postdoctoral Research Scientist гэж байгаа. За 1994 онд бол Монгол Улсын их сургуульд физикч бакалавр, дараа нь 97 онд дахиад мастер хамгаас 17 онд PhD Texas A&M University PhD хамгаалсан. За тэрнээс хойш бол энэ сургуульдаа дахиад постдокторийн судлаач хийж байгаа. Олон жил судалгааны ажил хийсэн туршлагатай физикч юм хүн байгаа. За тэгээд би бол өмнө нараад багштай холбоотой өөрийнх нь судалгааны талаар ингээд сонирхоод өөрөө надад бас ингээд айгүй тайлбарлаж өгсөн. Тэр судалгааны ажил нь бол айгүй сонирхолтой гой судалгаа хийдэг юм лээ. Гол нь нөгөө энэ Раман микроскоп ашиглаад тэр биологийн молекул за мөн одоо тэр макромолекул гэж ярьдаг. Тэдгээрийн одоо interaction тэр нарийн бүтцийн судалгаанууд хийдэг их гой судалгаа хийдэг юм лээ. Тэр судалгааныхаа ажлынхаа талаар өнөөдөр танилцуулахаар ажлыг танилцуулна уу гэж хэлж байна.
Talk about today my recent publication we had in a PNAS paper, like level-free drug interaction screening via Raman microscopy. And then I'm just talk about what is this technique can do, and then what is the advantages of this technique. And of course, because I present this talk mostly the physicists or laser optic background people, and then they're always wondering about why not you study these proteins, but because we are just kind of away from, not many physicists study proteins, and then it's the kind of like startup thing. Because probably everybody here mostly you might have biology background, and then protein is like no question about why we need to study protein. It's just necessary, like part of us, and then part of being well-being, and then etc. And I think the biggest question here, protein is mostly is if the protein is normal protein versus the protein is not abnormal protein, is the kind of starting disease, certain kind of diseases in our body. And then of course we necessary to study them, what is the normal, what is not normal, etc. That kind of gives, once we collect as much as information about between normal and abnormal proteins, then that gives us the opportunity to develop some kind of drug or understanding what we needed for us in our body being healthy and normal. And I think when I started looking at the protein, I kind of realized that the proteins are most of the existing technique we are doing, like protein ligand interaction, starting drug development and stuff. Most of the existing technique are done with the dehydrated proteins. And here this image is from gyro electron microscopy image, but then seen only 6%, about 6% of the proteins are the dehydrated state, but then the rest of them is the biologically active. You can see the shape of the protein is very different. Then if you do something biologically active things, you attach to the ligand, and it will be attached very different position, and it kind of makes sense studying them in biologically active environment. That's our technique, is this thermostable Raman interaction technique, profiling technique, is that we are doing everything is PBS solution, and then the concentrations are very nearly physiological concentrations, etc. Now I'd like to compare with the existing technique, what is out there, then for this protein interaction technique, and then we can kind of divide them what is the sample type they can do, or the preparation needed and stuff. The one thing good about the Raman microscopy is kind of we don't really need any sample preparation. That's the kind of, and then everything can be in water. Water doesn't have big influence on the spectra. And then, of course, the resolution of detection, and then how much you can reproduce the spectra, or another thing is how fast you can do the measurements, etc. This is the non-optical, first-party non-optical techniques, like everybody familiar with NMR or X-ray crystallography or X-ray. The other technique is very popular technique, and optical techniques are like CD or surface plasma resonance, and then interferometry, and then surface enhanced Raman spectroscopy. Now I'm adding our technique here on trip in the bottom volume. The row you can see here, our technique is kind of like the previous techniques you're seeing, like no other technique is perfect, but then kind of our technique is kind of very versatile technique, and can be stand up with these existing techniques. And why this is the reason why I call this technique thermostable Raman interaction profiling is because we actually actively cooling the protein samples on the sample filter, and we use the commercially available Raman microscope. I custom made the special cooled stage, and then I just feed it into the commercial Raman microscope equipment. Then what I did is I taken is the low concentration protein sample, like just only 10 microliter, and then usually I take the protein itself, ligand itself, and then I mix these two ahead of time, and then I did 24-hour incubation, and then I take the drop, 10 microliter out of the drop, and then I take the Raman spectra, and then after I take the Raman spectra, I can do the PCA, apply the PCA, see if the protein and ligand mixes are really moved to the clusters are moved from the protein. Because you can see here Raman spectra outputs number 2, the ligand spectra, the intensity is very, very low, but then proteins are over-dominated for this spectra, the Raman spectral spectral outputs. And here I would like to show you what is the new of this Raman being here like more than 50 years. What is the new here is you can see here the previous Raman studies, they're not really have a statistical representation of it because of they really having a hard time reproduce the spectra. Here, example of it is if I stop cooling, the first top part of the 4 spectra is the first one is like cooling, and you stop the cooling, then you can immediately see the spectra is the change a lot, and then you can see in the certain, like for example, looking at amide 1 region, the secondary structure is you can see the protein is degrading, and then the secondary structure is disappearing on this spectra without cooling. That's the main reason is people having a hard time reproducing the Raman spectra. And then if we do the active cooling, continuous cooling, we can able to reproduce the spectra without degrading protein sample. That's the main take for this technique. And this is the folklore of the trip technique, because the disease, if you have disease, then you have target protein or target antigen, and then you can have you can easily do very fast testing for the drug pool or antibody pool to really discriminate which one is the binds with the target antigen or target protein. Then you kind of bring out this one is very fast selection of the which one is the possible drug, which one is the possible antibody. But this is the kind of shows this technique is very versatile, of the size wise, because some of the existing techniques is only for certain sizes, but our trip technique seems works any size. And let me demonstrate the trip technique. And now first part application we did is two protein and small drug systems. And then, of course, first system was the streptavidin and biotin system. Then bacterial protein streptavidin and a small molecule biotin complex is one of the strongest binding partners occurring in nature. This is a very strong binding. Because of that, they used a lot of model like study high affinity protein ligand interaction, and their binding is used so many different biochemical sensing applications, and it's like I think it's a very very popular complex. And then you can hear for my Raman spectra, we can hear top as just shown the X-ray crystallography of their binding site. And then you can see here, down here, like the biotin with the streptavidin is the red color. The residue streptavidin binding with the biotin is the hydrogen bonding is here. Then one can see because of we have the limitation of our CCD, we need to have like, we had to divide the entire fingerprint region for 3 regions, the spectral regions. Then we did 3 PCA plots on each spectral regions. You can see here, Then the first region, we are seeing very good the separation between, you can see here the mix. The mix is the mixed cluster moved away from protein streptavidin clusters. Then you see the here the tryptophan 870 Raman peak is the strongest affected on the strongest binding between the biotin. But then we're looking at this is actually 4 of this tryptophan residues are interacted with this binding with the biotin. But it's not like hydrogen binding, but just the interaction happened between them. And then tyrosine 43 of the streptavidin, the hydrogen bonded with the biotin. We can see the hydrogen tyrosine's Fermi double vibrational mode is changed next for after the tryptophan. And then we can see our technique can see their binding very clearly here. And system 2 was the trans-thyretin where the 2,4-dinitrophenol is like a DNP and a complex. And then the TTR is the relevant to the important diseases such as Alzheimer, and its disease-causing action is the polymerized and form insoluble amyloid fibrils under certain conditions. And the DNP is the one of the inhibitor drug that can stop this polymerization. Here we can see the X-ray crystallography of their binding site, and then here I try to pull it out for the DNP molecule, how they interacting with the TTR residues. Then we are looking at the when I take the Raman spectra of it, then I'm looking at which regions are most changed, the mixes, because I tasted for the different hours, like just after mix, and then waited 2-3 hours, and then 24 hours. Then I would like to see what kind of changes we observe. Then first vibration, the modes we observe with the binding is the in-plane binding modes at 839. That's the in-plane is the phenol ring of the DNP with the NO2, these 2 molecules there because of the binding with the TTRs, the residue L908, it's and then water. It's kind of like these modes are changed. Then it's actually the modes are shifted downward 6 wave numbers and then intensified. And then second, but then these modes are it's just changed immediately and then didn't change over time. But then the second stretching mode of 1326 is actually change it over time just after mix, and then 24 hours is actually these changes is strengthened, and then this mode is shifted down to the 6 wave numbers too. Then clearly 3-bits can see that binding, and then we can actually do because Raman offers, you can do the time-dependent binding of this protein and ligands. And then second application we did we had the 6 antigen antibody systems, and monoclonal antibodies offers a major advantage in drugs discovery, and it became easy to quickly make a lot of the new antibodies. But however, the process of assessing antibodies, the new made antibodies to find promising drug candidate is the time-consuming, expensive. That's why our technique coming in, because our technique is time-saving and cost-effective. Then fourth 3 system was the protein A, and we had the goat IgG, human IgG, mouse IgG, and then the protein A binds with the very special spot, the site where the antibodies, it's their like heavy chain FC part that binds it. And here I show where the protein A is binded. Then the protein A binds all 3 antibodies. You can see here Raman spectra first, and then you see the spectral regions of 3 regions of the PCA. Then all region you can see here, the mixes are moved, the mixed clusters are moved from the antibody cluster, and then you can see the changes are in red wave numbers. All these changes happen, multiple changes happen for this binding. If you have any question, you can ask me after my talk, what is this weak binding, but my paper has more information about that too. And then just what happened with the other one? My antibody. Okay, then we have 3 systems, 4 and 6. Then we had this COVID-19 receptor binding domain, RBD, we called, and with 3 different antibodies. We used the same antibody used in previous study, but then now our RBD molecule is going to be binded with the light chain of the antibody, is like hair of the antibody. You can see here the binding site here, up here. And then here they call the first goat antibody will not going to be bind with the RBD, but the human antibody will bind with RBD with a certain location. Yeah, I marked here, location is yellow like residue here. And then mouse IgG is actually binds with the RBD and neutralize the RBD further duplication. And here we have Raman spectra of that, and then you can see here the PCA plot, and then the mix of the non-binding mix are almost overlapped with the goat antibody cluster for all 3 spectral regions. But the binding mix or neutralizing mixes are moved towards to the RBD, moves away from the antibody clusters. And then again, you can see here a lot of the Raman modes are changed due to that. But then some of them is similar with the protein A, but then most of them is they're different, and then different, especially between binding and neutralizing mixes. We are seeing certain residues are only occur on the neutralizing mix, and then or only occur where the binding mixes. And then this is the kind of shows our trip technique. We can very, very sensitive for the binding, where actually the single amino acid sensitive certain ways, and then we see here not only amino acid, we can see the secondary structure changes, etc. And then more details in my paper. But from here, I kind of move towards what else we're applying for this application, this new technique. Application 3 is now the paper we are submitted to the journal. This is the can trip detect changes in quaternary structure. Now here is this COVID-19, SARS-CoV-2 main protease MPRO is the necessary for the interaction of the virus with host cell receptors during viral entry. It plays a key role in polyprotein processing and is active in only dimeric form. And then you see the monomeric MPRO is actually inactive. Now the developing drug with dimerization inhibitor of this protein is the promising therapeutic agent capable of preventing replication and proliferation of the virus. And here we did the three different concentrations. Then here we have mass spectrometry results, and then even the 1 micromole MPRO we still have dominated with monomer, but we still have certain amount of dimers. 5 micromole we will have mostly dimer, but then still we have some monomers there, but then 10 micromole we are the mostly dimer. We would like to test the trip if able to see the natural dimer in monomer in the different concentrations. Then we have here you can clearly see that we are able to see PCR result shows us that we can clearly distinguish between monomer and dimer, and then mix of that too. Then we have you see here these thousands peaks are one of the important peak. Kind of shows us the monomer and then dimer. If the monomer dimerizes and then it's shifted to the blue shifted for 5 wave numbers, it kind of shows the dimer signature here. The different spectra same shows the changes on the thousands, and then we are seeing some changes for the 600s here too. Next, the following for that, we did now ligand decision dimerization. Now we have 4 different ligands, and one of them was the Pfizer, the active ingredients of the Pfizer drug called the Paxlovid. And we tested this 4 drug added into our 1 micromole monomer dominated MPRO samples with just the 1:4 ratio, 4 of the ligands, 1 to the protein. And then one can see here the Raman spectra, the MPRO spectra is very dominated. And then you see the ligand spectra almost flat. If I zoom it out, you can see here certain peaks are certain ligands has a little bit smaller peak here. And then we can see here the mixes we did mix spectra. Down here you can see, since ligand doesn't have much Raman, then in addition of this mix, the mostly is the MPRO. If there are any changes debated from MPRO, then that will gives us that there's something about the binding due to the binding between this MPRO and ligand. Here PCR plot, you can see here we monomer. Then you have this 4 ligand mixes here. They all shifted from the monomer spectra. And then you see again the influential, like the peak we observed for natural dimer, they are observed in the ligand-assisted dimerization, same way. The wave numbers shifted to the same way, downshifted. And then we see the both thousands and around the 600s, both locations we are seeing. That kind of clearly shown us the dimerization happened due to the ligand added to the monomer. Then now we are moving to the 4th application. It's the paper is now finally reviewed the small methods. It's hopefully published soon. And then estimating amino acid composition and secondary structure of the unknown protein. This is just the final result of that. And then here what we have used the TREAP technique to actually estimating these the amino acid compositions and secondary structure of the unknown. This is the technique is kind of really like only the first non-destructive technique exists bringing out in the first time here, and then it just kind of like just opened up so many opportunities for us to do a lot of different applications with this. And then number 5, application number 5 we did is the sickle disease detection via TREAP. And we did the patient blood from the hospital, and then we just ran through that special column, special like filter, and then just the centrifuge. Then we just take out the hemoglobin sample. Then we're able to distinguish the normal and then the sickle cells hemoglobin. But then the problem was there are many techniques doing the hemoglobin, and then the sickle shape the hemoglobin, because when you deoxygenate, the shapes is changed. But then our detection was the oxygenated hemoglobin, and it was most of the sickle disease blood sample will have like almost like 90 to 70% normal hemoglobin, and only like 10 to 30% of them is the sickle. Then that's the kind of like sensitivity gets very hard to. They need special like things to do before they're tested for that. But then it seems like our TREAP is sensitive without even discriminating that. Okay, then now this 6th application is coming. We are now working on this one is the can TREAP study binary and the ternary complex formation. Now we always had the 2 molecules, like the one protein and ligand, and then now we adding 3rd molecule into introducing it. Then we studied the degradation of the protein, the process, and it seemed like the TREAP is working very well. And yeah, that's this is the summary of like what's the TREAP offer: label-free, non-destructive, minimal sample preparation, fast, stable, reproducible Raman spectra from proteins in their near native environment, predicting amino acid composition, secondary structure of unknown protein ligand instructions for wide size of proteins, and then it seems like we're sensing changes in secondary, ternary, quaternary structures of the protein. And thank you. Any questions?